I'm Dr. Fakir Raveri, and this is clinical microbiology, susceptibility testing, and systematic approach to infectious diseases. Here's an overview of the human immune system. We can think of the immune system as innate immunity and adaptive immunity. The components of innate immunity include physical and chemical barriers. So physical, you can think of the skin, which basically prevents pathogens from entering the body. And chemical barriers, you can think of components of the saliva, and perhaps most importantly, the gastric acids that basically kill pathogens. Other components of innate immunity include mucus, the complement system, and innate cells such as macrophages, dendritic cells, granulocytes, natural killer cells. In general, the innate immunity is non-specific or broadly specific molecular patterns. And the actual immune response is through cytokines and chemokines, which lead to inflammation, phagocytosis processes, the complement activation, as well as activation of effector cells. Now, innate immunity is extremely important because in healthy individuals with an intact immune system, many features constitutively are present. In other words, you don't really need prior infection or immunization in order to have innate immunity. And the response is immediate. So it takes typically minutes to hours for the innate immunity to kick in and fight infection. However, it only persists for a few days, so it doesn't really give you long-term protection or memory. That's where adaptive immunity comes in. So the components of adaptive immunity includes humoral immunity, such as B lymphocyte cells and antibodies, as well as cell-mediated immunity, such as T lymphocytes or T cells. So adaptive immunity is antigen specific to a given pathogen. So it's not very broad, it's very specific to specific infectious diseases. And the actual response is through activation, proliferation, and differentiation of antigen specific B and T lymphocytes. It involves secretion of antibodies that can fight pathogens, as well as effector activity of T lymphocytes, and of course generation of memory cells, which lead to long-term immunity. And these mechanisms get activated either through actual natural infection or through immunization. Now, the first time the body is exposed to a pathogen, the adaptive immunity will take several days to develop, and that's referred to as primary response. So primary is the first time the body sees that pathogen. However, once the immune system has adapted to that pathogen, the next time the body encounters that same pathogen, there will be a rapid response due to the memory that develops. So because of that, there is a long-term duration of adaptive immunity. Now, once the adaptive immunity clears an infection, there is a contraction after elimination of pathogens that typically happens in a few weeks after the infection is cleared. And what contraction means is essentially, through adaptive immunity, the body will develop a lot of antibodies. And then once the infection is cleared, these antibodies start to wear off. So the levels of antibodies after a few weeks go down to very low levels, and that's called contraction. However, because the memory cells remain, and there are memory B cells are the cells that make antibodies, so memory B cells will last months and years, and there are also memory T cells that will probably last for the entire lifetime. And so the next time the body is exposed to the pathogen, because of this long-term memory, the body is able to expand the antibody levels very quickly. Now let's take a look at the hematopoiesis process. We're not going to go into too much details on this. We just need to know what the end products are and what their roles are in the immune system. Of course, this all starts with the hematopoietic stem cells, and these stem cells essentially differentiate into different cells, and these are the cells that exist in the blood. So, in general, we have 3 different kinds of cells in blood. Of course, we have red blood cells. Red blood cells, another name for it is erythrocytes. We have platelets. Another name for platelets is thrombocytes. And then everything else that you see here are essentially white blood cells. And another word for white blood cells are leukocytes. So now, because white blood cells have an important role in fighting infections, we're going to go into more details of the different types of cells that constitute leukocytes. Essentially, anything on the right-hand side on this pathway are lymphocytes. So we have B lymphocytes, we have T lymphocytes, and we have natural killer cells. And then everything to the left essentially comes from myeloid progenitor. So these are basophils, eosinophils, neutrophils, mast cells, and monocytes. Now, when we measure white blood cells in the blood, we typically count how many cells are in a microliter of blood. So, the normal range is anywhere between 4,000 to 11,000 cells per microliter. Now, if somebody has low levels of white blood cell, that's referred to as leukopenia. And of course, the other end is if somebody has too much white blood cell, it's referred to as leukocytosis. And that typically happens when people have an infection. Not every infection will lead to increased white blood cell, but if it is a type of infection that requires white blood cells to fight it, the body will start making more white blood cells. So, a typical sign of an infection would be if somebody has elevated white blood cell or, more appropriately called leukocytosis. And that would be if somebody has a level 12,000 or higher. Now, here on the right-hand side, you can see the shorthand way of writing some of these lab values. So on the left hand side is white blood cell, on the right hand side is platelet, at the top is hemoglobin, and at the bottom is hematocrit. And this is the shorthand when typically clinicians, when they work up patients, this is how they write. So you don't have to write white blood cell, you just write numbers here instead of these labels. And typically, if somebody says white blood cell is 4, they really mean 4,000. They're not going to write 4,000 or 5,000. So they typically write, like 6.2 or, 13. So if somebody writes 13 here, they really mean 13,000. Now, if you know somebody has leukopenia and they write 50 here, they're really talking about 50 cells. They're not going to write 0.050. So you just have to use it in the context. The same with platelets. So, typically people ignore the 3 zeros at the end. Now, hemoglobin and hematocrits are more straightforward. So the numbers are not too large. They're not in the thousands. So when somebody writes here hemoglobin is, 14, they actually mean 14. And hematocrit is in percentage, so that one is easy. Now let's take a look at the 6 basic types of leukocytes. So eosinophils constitute about 1 to 3% of all white blood cells. Basophils and mast cells, less than 1% of white blood cells. Neutrophils are extremely important. Neutrophils are the primary mechanism of fighting bacterial infection. So when it comes to bacteria, neutrophils have a primary role. And that's why you will see a large portion of white blood cells, typically 50 to 70% are neutrophils. Now, an important concept when it comes to neutrophil is the concept of segs and bands. So what we mean by segs or PMNs or polymorphonuclear leukocytes or just polys is that these are essentially mature neutrophils. And then the immature neutrophils are called bands, so band cells. And essentially, when the body makes new neutrophils, initially they are bands, and then it takes time for them to mature and become segs. So normally most of the neutrophils circulating in blood are mature in the mature form, with the nucleus of the cell being divided or segmented. So that's where the name segs comes from. Now, during infection, because the body tries to make more neutrophils than usual, you will see an increase in the band or immature neutrophils, and that's typically referred to as bandemia or left shift. So left shift meaning the cells have shifted to the immature side. And so if you see more than 10% of the neutrophils in the band cell, that's typically called bandemia. So this percentage here is 3 to 5% of white blood cells are normally bands, and white blood cells, about half of them, 50 to 70% are white blood cells. So this 3 to 5%, you can think of it as typically 6 to 10% of neutrophils. So if you see more than 10% of neutrophils as bands, that's typically clinically significant. Now, other types of leukocytes include monocytes, and they actually progress to become macrophages, and those are 1 to 6% of white blood cells. And then we have lymphocytes and their derivative plasma cells, and a big portion of white blood cells are lymphocytes, and they have an important role in developing memory. But also they have a big role when it comes to viral infections, and of course they are also important in other types of infections, including bacterial and fungal infections. And lastly, we have dendritic cells, less than 1% of white blood cells. Now here are some more details about the function of each leukocyte type. In general, eosinophils have a role in allergic reactions, including allergic reaction to antimicrobial agents. They also have a role in fighting parasitic infections. Basophils and mast cells are also active during allergic response, especially anything with histamine release, and they have a role in inflammation. As I mentioned, neutrophils have a big role in bacterial infection, but they're also important in fungal infections. Macrophages are not only important in bacterial infections, but they're also antigen presenting cells, so they activate lymphocytes. And of course, the three types of lymphocytes. B lymphocytes are the ones that result in plasma cells that make antibodies. B lymphocytes themselves are also antigen presenting cells, so they can work with T cells. T cells, there are different types. There are CD4, CD8 that are most commonly important types of T lymphocytes. They are not only important in developing long-lasting memory to bacterial infections, but they also have an important role in fighting viral infections. We have natural killer cells, and lastly we have dendritic cells that are also antigen presenting cells. Please pause this video and review the function of each of these leukocyte types. Now that we have a good foundation, let's take a look at our first learning objective. Given the results of a specimen stain, list the most likely organisms present in the specimen, and the specimen would be from the site of infection. So this could be blood, if somebody has infection in the blood, referred to as bacteremia, or could be urine, or sputum or CSF, or other types of specimen. Let's take a look at different types of bacteria. So in general, bacteria are divided into different groups based on how the cell wall is stained. So if you look at bacteria under microscope, you're not going to see anything because they're transparent. So in order to see them under microscope, we have to use some techniques to stain them so we can see them. If we look at bacterial cells, you will see inside the cell, there is a genetic material, and then the cell is surrounded by a cytoplasmic membrane, or the cell membrane. And then what you will see outside of the cell membrane is a cell wall, which is essentially made of peptidoglycans for most of these bacteria. And that's what we stain. So we stain the cell wall, and the cell wall is what gives the shape of the bacteria. So they will have different shapes. So, either rod shaped or spiral or other shapes that we will talk about shortly. And one stain that we can use is called the Gram stain. So Gram was, is the name of the person who came up with the technique. And some of these bacteria, when you use the Gram stain, they will turn blue, and we call that Gram positive. So something that's particular about Gram positive is that they have the cell membrane, and then outside of the cell membrane they have a thick layer of peptidoglycan, and then this layer gets this is the cell wall that gets stained blue. And then we have another group of bacteria that are called Gram negative. These essentially stain pink using Gram staining. And something that's particular about the Gram negative is that they still have the cell membrane followed by a thin layer of peptidoglycan. And the reason this is thin is that on top of the peptidoglycan there's a second outer membrane. So there are two membranes. So that's a major difference between Gram positive and Gram negative, that Gram negatives have this outer membrane. So the peptidoglycan is thinner. And then we have a third group that just doesn't get stained using the Gram technique. So then we will have to use a different type of staining. So the acid-fast staining is used. A good example is mycobacteria. For example, if you think of tuberculosis. So these get stained with acid-fast. And then the fourth group is what we call atypical, because they do not have cell wall. So, they do not have peptidoglycan, because these three have peptidoglycan that can get, one way or another, they're going to get stained. But the atypicals, because they don't have a cell wall, they don't get stained. So, any of these techniques you use, they will not be stained. So these are atypical. So these are in general the four types of bacteria that are clinically important. Now let's take a look at some of these staining and microscopy techniques. So as I mentioned, the Gram stain is the most commonly used technique, and this technique can stain bacteria, but also some fungi species can also get stained. Certain bacteria, most importantly mycobacteria, do not get stained using Gram stain, so acid-fast is what we use. And then there are specific techniques that are used when it comes to different types of fungal species. Let's take a look at taxonomy of bacteria. So taxonomy is essentially the classification of organisms in an ordered system that indicates a natural relationship. You may be familiar with Linnean taxonomy, so that's the name of the person who came up with this system, which ranks organisms by domain, kingdom, phylum, class, order, family, genus, species, and subtype of species for some species. For example, E. coli, Escherichia coli, is classified here, and the most useful in general is the family, genus, and species. And occasionally, for example, when it comes to E. coli, it is useful to know the order as well. And the reason is there are a few clinically important organisms that belong to the family of Enterobacteriaceae, such as E. coli. And recently, some of them were reclassified to different families. So we used to refer to these by the family Enterobacteriaceae. And because some of them are no longer in this family, it is clinically useful to refer to them by the order Enterobacterialis. Now, a strain is a subgroup within the species that has unique characteristics that distinguishes it from other strains. For example, E. coli is the species, and E. coli O157H7 is one of the subtypes of E. coli, and that itself can have different strains. For example, these strains are identified by a sequence number. So that's an example of strains. Now let's take a look at some specific examples of Gram-positive bacteria. Now in general, whether it's Gram-positive or Gram-negative, these bacteria can be either aerobic or anaerobic. So essentially aerobic means that they require oxygen for replication. Anaerobic means that they can replicate in the absence of oxygen. And these definitions get more nuanced from microbiology standpoint, but for us, clinically, that will suffice to distinguish between aerobic and anaerobic. And then with gram-positive specifically, we can look at morphology. So morphology is basically the shape of these and how they group together when you look at them under the microscope. So essentially, we can see cocci, and this is essentially in circular shape, and they can be in clusters. So cluster means that several of them are clustering together. And then you can have cocci, which is essentially circles in pairs or chains. So pairs will be a couple of them, and sometimes that's referred to as diplococci. And then, if you have a chain, they don't cluster. It's just, just like a chain. And then we have rod shape that are called bacillus, and the plural for it would be bacilli. And I should say cocci is a plural. The singular form is coccus. So if you're talking about the single bacterium, it will be a coccus. And then so for gram-positives that look like cocci in clusters, typically there's a test they do, then these tests catalase positive. And then on top of that, in the micro lab, they can do a coagulase test. And if you have coagulase positive, then you have a very important, clinically important organism, Staphylococcus aureus. Okay, and the reason we're talking about this is because it takes some time to identify what the organism is. And if somebody has an infection and we don't know what it is yet, but they look at it under the microscope and they say, well, it looks like cocci in clusters, and we also did a coagulase test and coagulase test was positive, as clinicians we can anticipate Staphylococcus aureus, so we can give appropriate treatment. So that's why we're covering this. We're not trying to become microbiologists, but as clinicians you can use this clinically to help patients. And if they say the coagulase is negative, you have several options, and together all of these are referred to as coagulase negative staph or coagulase negative staphylococcus. And there are several species that are coag negative, and I have listed 4 that are more common. And Staphylococcus lugdunensis is one that can cause severe infection, so that's why I have bolded it. But so these are typically present on the skin. The same with Staphylococcus aureus. All staphylococci are typically present on skin, and occasionally they find their way into the body and cause infection. And then for the catalase negative group, which they do not look like a cluster, so they will be in chain or maybe pairs. Then we can do the hemolysis test. So what a hemolysis test would be is that essentially an agar is essentially a plate. So what we do is that we use sheep blood, and we basically let these organisms grow and see what they do to the blood cells. So the beta hemolytic group, which you can see in this picture, essentially they lyse blood cells, so they will break it down and it will basically clear the blood, and that's called beta hemolytic strep. And the alpha essentially reduce hemoglobin, and they cause this greenish color. And then there is a third group that don't really do anything to the blood, and these are referred to as non-hemolytic because they didn't break it down. So essentially that's how we group these. So beta hemolytic strep, alpha hemolytic strep, and non-hemolytic. So in the beta hemolytic group, we have 2 important Streptococcus species. So Strep pyogenes, which is sometimes referred to as group A strep. And then we have Streptococcus agalactiae, which is referred to as group B strep. So these are important, and you should know these interchangeably, because sometimes people may refer to to them as group B strep. So they might say, okay, for this patient, we are trying to cover group B strep. So you should know that they are referring to Streptococcus agalactiae. And if they say we want to cover group A strep, you should know they're talking about Streptococcus pyogenes. And then for the alpha hemolytic group, the most important is Streptococcus pneumoniae. This is very important because it causes respiratory infections, and we'll cover this in detail throughout the course. And then there's also the viridans group Streptococci. When it says group, there are multiple species, and there's really no need to know specific species. They're collectively referred to as viridans group Streptococci. And then the non-hemolytic, we have two Enterococcus. In the old days, these were also called Streptococcus, but nomenclature has changed over time. So nowadays, these are called Enterococcus, but they're also in the same group as the rest of the strep. Enterococcus faecalis and Enterococcus faecium, these are two organisms that are kind of the frenemy, so they sometimes they are good because they can be as part of the flora in the body and do some good, and sometimes they can be bad and they can cause an infection. So that's why they're tricky. So you have, and we'll cover this throughout the course. So sometimes, for example, they might be in the urine and prevent urinary tract infection. Other times they might be the cause of urinary infection. So it becomes tricky when we identify this in a specimen. So a lot of caution should be used. And then lastly, these are just the gram-positive bacilli, so Listeria, Bacillus anthracis, and Corynebacterium diphtheria. Now let's take a look at gram-negative bacteria. Again, we're focusing on aerobic for now. So you will see that majority of gram-negative are actually rod-shaped. So these are bacilli. And what we do with gram-negatives is that we do a lactose fermenting test. So if they can ferment lactose, we call them, actually the important thing is to identify what's not fermenting lactose, and they're typically referred to as non-fermenters, okay? But if they do ferment lactose, then, they do oxidase tests, and, for this course, you don't have to worry about the oxidase test, but know that when it's lactose fermenting, for the most part, we're talking about Enterobacterialis, and Enterobacterialis is a family of several organisms, and Enterobacterialis used to be called Enterobacteriaceae. And the important organisms in Enterobacterialis family is Escherichia coli or E. coli, Klebsiella species. So when it says SPP, that means there are multiple. So for example, there is Klebsiella pneumoniae and there is Klebsiella oxytoca. And there is Enterobacter cloaceae. There are several Citrobacter species. There are Serratia species, Providentia species, and Protease species. So these are all under Enterobacterialis. And then we have, other lactose fermenters, so Aeromonas, Pasturella, and Vibrio species. And then when it comes to non-lactose fermenters, and this is bolder because these are very important organisms because they typically are very resistant to a lot of antibiotics. So the most important would be Pseudomonas aeruginosa. So this causes a lot of severe infections and becomes difficult to treat. And also Acinetobacter baumannii, which is less severe than Pseudomonas but can be more resistant to a lot of organisms. And then, Stenotrophomonas and Burkholderia also can be very, very resistant. And this typically, Stenotrophomonas and Burkholderia typically more common in immunocompromised patients. And then for the cocci in the gram-negative group, we have Neisseria meningitidis, Neisseria gonorrhoeae, and then we have Haemophilus influenzae and Moraxella catarrhalis. These two are actually kind of between cocci and bacilli, so they're referred to as coccobacilli. So the shape under microscope is between a circle and a rod. And then spirochetes, which are essentially look like a spring. So we have a couple of them, Troponema pallidum, as well as Borrelia burgdorferi. Now let's take a look at some anaerobes. So anaerobes, gram-positive is Peptostreptococcus, and then we have a series of Clostridium and Clostridioides difficile. So Clostridioides difficile also used to be called Clostridium difficile, and nomenclature changed a few years ago. And we will cover these later in the course. And the gram-negative anaerobes, essentially the most important you need to know is Bacteroides, because any intra-abdominal infection, Bacteroides have an important role. Atypical bacteria include chlamydia species, including chlamydia pneumoniae. That's not a sexually transmitted infection. It can cause, pneumonia. And then we have the sexually transmitted chlamydia. Also, we have Mycoplasma and Legionella. And then with the acid fast, we have Nocardia species, we have Mycobacteria, as well as Actinomyces. Looking at the results of a specimen stain, we listed the most likely organisms present in the specimen. Now let's describe the basic process of microbiology testing for identification and susceptibility. Let's take a look at the process of microbiology testing when a patient who's suspected to have an infection presents. So when a patient presents with symptoms and signs of an infection, we'll call this time 0. And typically, it will take anywhere between 1 to 3 hours for clinicians to examine the patient and, if necessary, collect specimens. And these specimens will be from the site of infection. For example, it could be blood or urine or any other specimen. And then the specimen will be sent to the lab, to the microbiology lab for testing. And what happens at the microbiology lab is that you typically have to incubate these specimens in order for bacteria to grow before we can detect anything. And this can be done using automated systems, or it can be done on an actual plate. So these plates are called agar plates. And what they are is that they essentially have nutrients in them for bacteria to grow, and you just let them sit there and incubate under the right temperature for bacteria to grow. And then after a while, typically anywhere between a few hours to a few days, depending on the organism, we may start to find colonies of the organism. Now, of course, it's possible that nothing may grow. So, sometimes it will be difficult to identify. But if there is a specific organism that will be identified, it will be growing typically between 12 hours to 5 days. And depending on what type of infection we are suspecting in the patient, if we think that it might be an organism that's a very slow grower, it will be wise to keep the specimen for beyond 5 days in order to give it enough time to grow. Now, if something does grow, that's when, typically they start to stain it and then look at it under the microscope. So while the process of staining, is not time consuming, now if you look at the entire process, it typically takes somewhere between, half a day to a day, if not more, before they can actually look at it, stain it, and identify, if it's bacterial infection, whether it's gram-positive, gram-negative, or atypical, and then look under microscope to look at morphology. And then there are a series of tests that will be done in the micro lab in order to identify. So this is where they do those, the lactase test and oxidase test and hemolysis test. And so there will be numerous tests that will be done if applicable in order to actually identify what the exact organism is. And that typically takes anywhere between 1 to 2 days after staining. So now, this is typically day 2 since that patient presented at the minimum. And once an organism is identified, so for example, if they say this is E. coli, then the next step would be to identify antimicrobial susceptibility. So then, it typically takes another 24 to 48 hours to do various testing. And we will go over some of these susceptibility testing. But the key point here is that it will take, another day or two after the organism is identified in order to find susceptibility results. And what that means is that, they will tell you like which antibiotics will be active against this organism and which ones will not be active, so that we can streamline treatment. Now, as technology advances, we have rapid diagnostic tests that can expedite this process. So, when we actually grow an organism, we can directly put the some of these colonies into rapid diagnostic tests, and these rapid diagnostic tests will be able to very quickly, in a matter of anywhere between under an hour to just 2 or 3 hours, tell us exactly what the organism is and what the susceptibility results are. Now the problem is these diagnostic tests are limited to only a select few organisms, so they're not comprehensive of every possibility. So it's not a replacement for the traditional process. So even though we use a rapid diagnostic test, we still need to do the traditional identification and susceptibility testing, because in case the patient has an organism that is not included in this rapid diagnostic test, we still need to figure it out, and you don't want to slow down this process while the rapid diagnostic test is processing. But certainly these rapid diagnostic tests are advancing very quickly year after year. Now, what happens in parallel to this process is what we do to treat the patient, because we're not going to wait for this process to complete and then start treating the patient, because the patient could die by day 3 when we find out exactly what antibiotic will work. So what we do is initially we do empiric antimicrobial therapy. So what empiric means is that we don't really know what's causing the infection, but we will do our best guess to, based on the signs and symptoms, we can anticipate what organisms are possible, and we can use antibiotics broad enough to cover the possibilities. So now we can kind of look at the timeline of treatment. So we typically, we want to avoid treatment as soon as the patient arrives, because if you give the antibiotics immediately, you might not be able to identify anything because you kill all the organisms. So you want to get your samples first if possible, and then start treatment after you get the samples. And then, once we get rapid diagnostic results, that's your first opportunity to optimize treatment. So based on what you find, you may be able to change the antibiotics to have something that's better for the patient. The same with, when you have gram stain results, you can change the antibiotics. And ultimately, when antimicrobial susceptibility results are available, at that point, we will switch from empiric therapy to pathogen-directed therapy, meaning that we know exactly what is the best antibiotic for the patient that will be active against the organism and will be the narrowest that would get to the site of infection and will result in the optimal outcome for the patient. When it comes to phenotypic susceptibility testing, the concept of MIC is extremely important. So MIC stands for minimum inhibitory concentration, and it is the lowest concentration of an antibiotic that inhibits visible growth of a microorganism. And that's why, we say a phenotype is what actually appears. So, it will be visible to the actual human eyes. And that's to distinguish it from genotypic susceptibility testing. So this is, regardless of what resistant genes may be present in an organism, the minimum inhibitory concentration just shows you what's the minimum concentration to inhibit growth of organism. And there are different ways to measure MIC. So the gold standard is the broth microdilution, and by gold standard I mean this would be the reference method, and any other method would be something to as a surrogate for the broth microdilution. And here's how broth microdilution works. So you can kind of imagine a series of test tubes in the microbiology lab, and then in each test tube we will have a broth. So what broth is essentially nutrients for bacteria to growth. Now these are sterile, so there are no bacteria in them, and what we do is that we put antibiotic of choice that we are trying to test in these test tubes, and we increase the concentration of antibiotic in each test tube, so kind of double the dilution. So essentially one of them will not have any antibiotic, so the concentration would be 0, and then we kind of double the concentration in each test tube, and then we inoculate these test tubes with the organism that we are trying to test, and we let it grow. And then after the incubation period is over, we can see that it actually, the growth becomes visible. So you can see that it has become cloudy, or the color may have changed. And then you can see at what concentration nothing grew. So for example, in this example, you can see that at the concentration of 64 and above, nothing was growing. So the minimum concentration was needed to kill the organism in this example is 64, because a concentration of 32, the organism was growing in the test tube, but at 64 or higher, nothing was growing in the test tube. And that's how they measure the minimum inhibitory concentration for a specific antibiotic. And MICs, so every antibiotic has a different MIC for each organism that it covers. So if you have antibiotic A, it will have MIC for E. coli, let's say. It will also have a different MIC for Klebsiella, and so forth, if the antibiotic A is active against these organisms. And then the same when you go to a different antibiotic, you have to measure the MIC for that different antibiotic for each organism. Now, another method to do is disk diffusion, also referred to as Kirby-Bauer method. And the way this works is that essentially, instead of test tubes, which you can imagine that it will be very labor intensive, so broth microdilution takes a lot of space and requires a lot of labor, so it may not be feasible. And that's why we say it's a reference method. So a much more feasible method would be disk diffusion, and here's how it works. So instead of test tubes, you have an agar plate. So again, this plate is sterile. What's on the plate is nutrients for bacteria to grow, and what we do is that you inoculate the agar plate with the organism that we're trying to test, and then immediately after we inoculate the plate, we place several discs. So these discs are essentially different antibiotics. So each disc has a certain concentration of the antibiotic, and when you place these discs in the region near each disc, this antibiotic starts to diffuse, and the further you go from the center of the disc, the concentration drops. Okay, and then at some point when you go too far from that disc, that specific antibiotic may not be present. So each disc has a different antibiotics. So you are simultaneously testing multiple antibiotics for this particular organism. And then if you have multiple organisms, then you will have one plate for each organism. So you can have several plates set up. And then you let it grow. So after the incubation period, you kind of can see that there are there is essentially a lawn of the organism that grows, And if the organism is susceptible to that antibiotic, there will be a zone of inhibition. In other words, because antibiotics have diffused in this region, the organism will not be growing. And if it's resistant to it, you can see that the organism will grow through the disc. Essentially means that that antibiotic was not active. And then we can kind of correlate this to some, whether it's, is it really susceptible, or is there some sort of intermediate resistance going on? So we can measure the zone of inhibition. So you can see this is a circle. They can actually measure what's the diameter of this zone, because as you can see, some of them have a smaller diameter, others have larger diameter. So the larger the diameter means the more active the antibiotic is. And we'll talk about how to interpret this zone of inhibition shortly. And of course, if it goes through the disc, that means that the organism is completely resistant to that antibiotic. So that's disc diffusion. And then another method, this diffusion only tells you if it's susceptible, intermediate, or resistant. It doesn't really give you MIC. Okay, so broth microdilution will give you MIC, and then based on the MIC, we can decide if an organism is susceptible, intermediate, or resistant to the given antibiotic. This diffusion only tells you if it's susceptible, intermediate, or resistant without the MIC. So another way to do it in order to get the MIC is the e-test. So the way e-test works is similar to disc diffusion. The difference is that instead of placing discs and then measuring the zone of inhibition, you put a test strip. So there is a strip that has a given antibiotic at different concentrations. So you can see that, at the top, it has a very high concentration of antibiotics, and the more you go down the strip, the concentration drops. And then, again, you inoculate the plate, you place the strip, and you let it incubate. And then after incubation, you can kind of see a zone of inhibition. And then you can see at what concentration the antibiotic stopped inhibiting growth. So that would be your MIC. So in this example, we can see that it stopped growing at concentration of 0.126 or higher. So the MIC would be that number, 0.126. So that's something that's also done in practice. And another way that's more commonly used is the automated machine. So there are certain machines that are automated, essentially. So you can see the one example in this picture where you essentially load this with your sample. You can test a lot of samples simultaneously. So there are these cartridges that you put in, and you just run the machine, and it may take some time for the machine to run, and then in a matter of an hour or so, and then it will actually give you MICs. So then you can get your MIC and interpret it as susceptible, intermediate, or resistant. And this is, these automated systems are most commonly used for majority of the samples. Now the issue is that because these machines are difficult to make, they typically come preloaded with certain antibiotics. So if a new antibiotic comes to the market, it may not be available in this machine, and that's where e-test comes in. So anytime a new antibiotic comes to the market, the manufacturers will make e-tests available so that micro labs can test for susceptibility so they can use the new antibiotic. Now, when you get your MIC results, or if you get results from this diffusion, what they really tell you is, those MICs are essentially in a test tube or on a plate, which we call in vitro, meaning it's not inside a living host. Although we know there is a minimum inhibitory concentration, that doesn't necessarily mean that we would be able to achieve that concentration inside the patient's body. So, it has to do with, the dosing of antibiotics that are safe would we be able to achieve that concentration in the patient's body. So in order to interpret that MICs and see if it's feasible to use that antibiotic, there are criteria. So just knowing the MIC doesn't really tell you whether you can use it or not until you interpret it. And the way we interpret it is that we use CLSI is the organization that gives us the interpretive criteria or breakpoints for MICs. And and I will show you how to navigate this document. But essentially, they will tell you for a given organism, for each antibiotic, they will tell you at what MIC cutoffs it will be considered susceptible, intermediate, resistant, and for some of these, there will be a susceptible dose dependent, meaning that it would be susceptible as long as you give a certain amount of antibiotic. So if you click the link, you will go to CLSI's website where they will host the M100 document. The M100 document is the one that has interpretive criteria for all antibiotics. So it doesn't have antifungals or antivirals. It's specifically gram positive and gram negative organisms. And then this is available for free, so you can just click to enter as a guest. And then you just make sure you click on M100 document, and this gets updated annually. So, for example, next year you will see the 2023 edition. So, keep that in mind. And then, once you go here, you can use the table of contents, and there is a table for each organism. So if you scroll down, you will see that starting with table 2A. So this one 2A is for Enterobacterales, and then the next table is for Pseudomonas. The next one is for Acinetobacter. Next one is so on and so forth. So if you are looking for breakpoints for specific organism, we're going to use Enterobacterales for this example. So if you click Enterobacterales, it will take you to the page for Enterobacterales. And this document is intended for microbiology lab staff. So, a lot of this you can ignore. The part that you need when you look at the breakpoints is the table. So you will see that it has columns for susceptible, dose dependent, intermediate, and resistant. And there are two sets of them. So one of them is based on MIC. So if you are using an automated machine that gives you MIC, or if you're using e-test, this is the part you will use. And if you're using this diffusion where the diameter of zone of inhibition is important, they will tell you, how big does the zone need to be. So in this example, let's say if you have E. coli as the organism and you're testing against ampicillin in this diffusion, they're telling you that if you measure the diameter and if at least 17 millimeter, it will be susceptible. Now, if it's between 14 and 16 millimeters, it will be intermediate. And if the zone is smaller than 13 or less, it will be resistant, including, if the zone is 0, which means that it just grew through the disc. Now, you guys don't need to memorize any of these numbers. I'm just trying to show you how to navigate and use this document. Now with MIC, so again for susceptible, it will say MIC of 8 or less is susceptible. So in other words, if you test it and MIC comes back and it says MIC is 4, you look at here and say, oh good, it's 8 or less. So, for that patient, it would be susceptible. But if you had, another test result come back and said the MIC is 16, you look here and say, oh, it's 16, so it's not susceptible and it's not resistant and it's intermediate. And what intermediate means is that if you're using the usual approved doses of ampicillin, it will likely not work. But if you were to use higher doses, it will probably work. And if it's resistant, doesn't matter. If you use the maximum dose that's safe in patients, it's just not going to work. So you do not want to use ampicillin if it's resistant. And so if the MIC comes back and it's 32, it's resistant. If it's 64, it's resistant. Now with intermediate, also in general, if something is intermediate, we try to avoid it if we have other options. So occasionally, you may not have other options available. So in situations like that, we will have no choice but to use it if it's intermediate. But if it's resistant, under no circumstances will we be using that antibiotic. In this case, susceptible dose dependent doesn't exist. So susceptible dose dependent only exists for certain antibiotics. So for example, a good example is cefepime. So if you go down to cefepime, which is a cephalosporin, you will see that under the column for susceptible dose dependent, they have listed 4 to 8. So this means that if you have, for example, Klebsiella pneumoniae, which is a member of Enterobacterales, and you get an MIC for cefepime back and it's 2, you're going to say, okay, good, 2 is susceptible. Now, if it comes back and it's 8, then you're going to look here and say, oh, it's 8. It's not susceptible, but it's susceptible dose dependent. And then you can look at the notes here. So what does that mean, dose dependent? And then they will tell you that essentially, if it's susceptible, it's based on if you're using a dose of 1 gram every 12 hours. But if it's 4 or 8, you need a higher dose of higher than 1 gram every 12 hours. And they guide you to Appendix E, where they have a specific recommendation. And essentially, you will see in that part that, if it's MIC of 4, you need to have 1 gram Q8 or 2 gram Q12. And if it's MIC of 8, you need to use the maximum dose, which is 2 gram Q8. Maximum, as far as FDA approved maximum dose. And of course, if it's 16, it will be resistant. And because there is a susceptible dose dependent, then there is no intermediate. So they have eliminated the intermediate for this antibiotic. So that's how you navigate the CLSI document. Here's another example. So let's say we have identified E. coli, and then we did broad microdilution to see the MIC. So, looking at this, you can see that this test tube, this is cloudy, and these 3 are clear. So, which means that organism E. coli was growing in these 2, but not here, here, or here. So the minimum concentration needed to kill E. coli in this example was 8. So MIC is 8. Now, just knowing the MIC alone doesn't tell you anything of what you can do. And I should say this is ciprofloxacin in these test tubes. So just knowing MIC is 8 doesn't mean if we can use ciprofloxacin or not. So what we need to do is to interpret this MIC. So CLSI document, I put a screenshot of the table here. You can see that for MIC, they say to be susceptible, it has to be less than or equal to 0.25, and then anything greater than or equal to 1 is resistant. In this case is 8, so it's definitely resistant. So this E. coli in this sample is resistant to ciprofloxacin. Even though, if you give high enough of ciprofloxacin, it's going to kill it, right? But it's considered resistant because we cannot given the safe doses of ciprofloxacin, we will not be able to achieve these concentrations in the human body. So we will consider resistant and not use ciprofloxacin for a patient who has this isolate. Okay, that was phenotypic testing. Now, when it comes to genotypes and testing for genes, we have rapid diagnostic tests, sometimes called molecular tests. And, some of the common ones is the PCR test, so multiplex PCR. There are different tests out there. So one example is the BioFire film array BCID2. So this test that was developed in recent time can detect any of these organisms listed here. These are the ones that are commonly encountered in clinical practice and are problematic. So it's good to identify these rapidly. Now, if it doesn't detect any of these, doesn't mean that the patient doesn't have an infection. It just means that it could be something that's not on the list. And of course, occasionally there could be a false negative, right? So, it could be that somebody has one of these and the test just couldn't detect it, and that's extremely rare because these tests have an extremely high sensitivity. And then the important thing about these PCR tests is that it can also detect these genes. So these are genes that we will cover in a different topic on another lecture. But if these are detected, that means that the organism can be resistant to certain antibiotics, and we'll cover what those antibiotics are later. And also, this is really fast. So this will give you results within an hour once you run the PCR. And of course, you have to wait, probably around half a day to a day for the organism to grow first. But once you have an isolate, then you put it in the PCR, and then within an hour, it will give you the results. Another test is the Verigene test that's commonly used. There are two different. They have one specifically for gram-positive and one for gram-negative. So these are, again, the organisms that it's capable of detecting and the resistant genes that go with them. And this one takes a bit longer, but still pretty reasonable. It will tell you the results in about 2 hours, 2 to 2.5 hours. Now, beyond PCR, we have other technologies. So, for example, MALDI-TOF, which is a powerful machine. It can virtually detect all bacterial and fungal organisms, and it's pretty fast. It can do it in 30 minutes. However, its limitation is that it cannot reliably detect resistance. Now, there is a question mark here because this is an area of active research, so it is under development. But currently, most organizations use this for detection specifically, not for resistance. Other technologies, Pinafish. So we have quick fish that's actually pretty quick, 20 minutes. It can detect select organisms, but again, it cannot detect resistance. And more recently, we have Accelerate Pheno test, which can detect a number of gram-positive and gram-negative organisms as well as Candida species. And this thing is unique because it can also report MICs. So in general, it takes about 2 hours. So it's still pretty fast. It can take about 2 hours to identify these organisms, and it takes about 7 hours to report, susceptibility results for select antimicrobials. And lastly, we have a combination of NMR and PCR. So we have the T2 bacteria. This one takes about 3 to 5 hours, and it can detect some organisms but not resistance. But this one is unique because it can actually work directly from whole blood. So if you can think of the process of identification and susceptibility, typically when we get the blood samples from the patient, they need to be incubated and see some growth before we can put this in rapid diagnostic test. With the T2 bacteria, essentially, once you get the sample, it goes directly into the diagnostic test. So that will save you half a day or so, in identifying the organisms. So that's pretty good with the T2 bacteria, and this is a relatively new technology, and more development will be under. We interpreted the results of a specimen stain to list the most likely organisms present in the specimen. We described the basic process of microbiology testing for identification and susceptibility. Now, let's identify the normal human flora and their role in infectious diseases. Now, normal flora are absolutely essential for health. There are non-sterile sites in the human body where these flora reside. However, there are sterile sites that are not supposed to have any organisms in healthy hosts. So in general, all internal organs are considered sterile. So they should not have any flora. For example, in blood, we don't anticipate to have any organisms. So if you do find an organism in this sterile site, they're considered in infection for the most part. So especially blood. If you find anything in the blood, it's considered infectious. The same with the central nervous system, cerebral spinal fluid, in the liver, spleen, kidneys, lower bronchi and alveoli, and the bladder for the most part. So the bladder is a bit tricky because Especially in young men, the bladder is supposed to be sterile. But as part of aging, for most people, as we get older, we start to have colonization of bladder. That may as people get older, bladder is no longer sterile. But a young man, it will definitely should be sterile. In women, it's common for women, given the anatomical differences between men and women, that the bladder will be colonized. And women, as they get older, also the percentage of colonization goes up. And anybody who has a urinary catheter almost certainly has colonization of bladder. So the bladder is kind of tricky on this list, but the rest of this list is pretty much, if you find an organism, it's likely due to infection. But let's focus on the normal flora in these other sites. So the skin in general has gram-positive aerobes, such as any Staphylococci species, including Staph aureus. So if you find Staph aureus on the skin, it's not necessarily infection in a healthy host. Occasionally, this may cause skin infection. But in the absence of any signs and symptoms of an infection, it's okay to have these organisms on the skin. That's part of the flora. There are obviously oral flora in the mouth. So, Fusobacterium, Peptostreptococcus, and Viridans streptococci. And then for nasopharynx, again, Viridans streptococci, Staphylococcus aureus. And the reason I have bolded some of these are the ones that are clinically important, so you should definitely know them for sure. And this is important because, so let's say somebody has an infection and you find Staphylococcus aureus in the nose, it doesn't often mean that it's an infection, so it could be part of flora, but if you detect it, you can kind of anticipate, because not everybody will have it as part of their flora. So, it's good to screen patients just to see if as part of their flora they had Staphylococcus aureus. So if you have infection in other parts of the body, you can kind of anticipate that Staphylococcus aureus found its way to other sites to find an infection. So perhaps the lower part of the respiratory system causing pneumonia, and we will talk about those scenarios throughout the course. Let's see. So the anaerobes in the nasopharynx are the same anaerobes as the mouth, and there are some gram-negative aerobes in the respiratory tract. So H. flu, Haemophilus influenzae, and Neisseria meningitidis is one. And then in the vagina, there are flora. For example, E. coli is a resident of vaginal tissue, and group B, Streptococci, and Listeria. Of course, when people mostly talk about flora, they typically talk about the GI tract, so gastrointestinal tract, commonly referred to as gut flora, and there are a lot. So you can see on the right-hand side that because of the harsh acidic environment of the stomach, not many bacteria can live in the stomach, and that's in general a defensive mechanism. And as you go down the GI tract, it's still acidic throughout, but the acidity decreases down through the small intestine and then the large intestine. So you can see as the acidity decreases, the number and variety of different flora increases. So especially in the large intestine, you will have a diverse body of flora, and this is a good thing, right? So the more you have these healthy organisms in your gut, they prevent through competition other problematic organisms from growing. So you're less likely to have an infection in the GI tract. Most commonly what we worry about is Clostridioides difficile infection. So because these organisms will compete with C. diff, it's healthy to have a healthy number of flora here. And this is also the reason why we are very careful when we choose antibiotics to treat other infections, because pretty much majority of antibiotics will affect the normal flora. So we want to avoid unnecessary antibiotics in order to preserve the flora. And when we cannot avoid antibiotics, we want to make sure that we use them for the shortest amount of time in order to minimize the collateral damage caused by antibiotics. But so you can pretty much see that these are mostly Enterococcus faecalis, which resides in the GI tract. Different species of Clostridium. A lot of these are anaerobes because this is an anaerobic environment for the most part. But some aerobes can survive here too. So Enterococcus is definitely aerobic and can have a big role here. As well as members of Enterobacterialis, so E. coli and Klebsiella certainly reside here. And the most important anaerobe is perhaps Bacteroides that resides in the lower GI tract, but pretty much unlikely to be found in other parts of the body. And in general, when people talk about antibiotics and ask if an antibiotic has anaerobic activity, they're primarily talking about Bacteroides rather than other types of anaerobes. Now, some important clinical concepts for interpreting cultures is, colonizer and contaminant. So anytime you have something growing in a culture, you need to ask yourself, is it coming from the source of the specimen, or did it get contaminated in the process when it was being collected? So a contaminant will not be from the site of the specimen. It could be due to improper technique, and then the colonizer is, if you detect something in your sample and it's not a contaminant, it means it came from the source of the specimen, but maybe perhaps it was not causing an infection. So a colonizer is essentially part of the flora, or if they didn't normally reside in the site as flora, but colonize it and it's not causing any problems. So that would be a colonizer. A good example is, some people may have Pseudomonas in the lungs, for example, and it may not, if they don't have any signs and symptoms of infection, it would be just a colonizer that normally would not be there in a healthy person. But, for example, someone with cystic fibrosis may have colonization of Pseudomonas in that region. Let's take a look at some terminology of human flora. So the first concept is microbiota. So microbiota refers to the collection of all living organisms present in a specific ecosystem or environment. So for example, the gut microbiota refers to all living organisms living in the human gut. And while dominated by bacteria, microbiota does include fungi, phages, archaea, and protists. And sometimes you may hear the term mycobiota, and that's specifically referring to the fungal component of microbiota, which is an emerging field in microbiota research. The next concept is microbiome. So this is essentially the combination of the microbiota as well as all the genes that come with these organisms. And because these genes, when translated into their product, can have an effect on the host, it is important. And to give you a perspective, the human genome has somewhere about 23,000 genes. So that's what's in our DNA. But the genes that come from the microbiome collectively is somewhere between 10 million genes to 200 million genes. So depending on an individual. And the last concept is the virome. So these were all living organisms, and since viruses are not living organisms, there could be viruses in the gut or as part of the flora, and not just viruses but also phages. And for most intact immune hosts, these will not cause an infection, so they will essentially be part of the flora, and they may have an effect on the health of the host. And that includes phages as well as viruses that infect other organisms such as archaea, but as well as viruses that could potentially infect the human cells when there is an immunocompromise. Now, it's important to understand the concept of prebiotics, probiotics, and postbiotics. So prebiotics comes before the probiotics. So prebiotics essentially the food for probiotics. So this is the food for bacteria that are part of the flora. And for the most part, this is essentially fiber, but also could include other substances that are used by the bacteria. And probiotics, of course, is the live microorganisms which, when administered in adequate amounts, confer a health benefit on the host. And probiotics could be part of food, but also could be available as pharmaceutical products. And last but not least, there is the concept of postbiotics, and this is essentially the byproducts made by these probiotics that confer the health benefit. So essentially, if we think that probiotics are good for you, the mechanism by which they are good for you could be pathways that are activated by some of these postbiotics. Now, in human flora, there are 5 phyla that essentially make up the flora. So these are in order of decreasing abundance. So the most common phylum is the Firmicutes or Firmicutes, some would say. The second one is Bacteroidetes. The third one is Proteobacteria. Fourth one is Actinobacteria. And in recent time, we also have Microbiology. And essentially, these are the 5 phyla. Now, you can see that when it comes to genus, there are many there are a lot of them in each of these phylum. The ones that are bolded are specifically the ones that are available as probiotics in the United States. So you will see that most probiotics products will include Lactobacillus species, and Lactobacillus has many different species that are not listed here. Occasionally, some products may include Streptococcus and Clostridium, so you just have to read the label to see if they include them. Currently, there aren't any probiotics products on the market that have Bacteroidetes. Some products have Bifidobacterium, and again, Bifidobacterium has many different species, so you will have to see if they're included. And in recent time, there is one product on the market that has Akkermansia available. When it comes to microbiota and probiotics, it's very tricky, so it's really hard to find for sure, like which probiotic would be beneficial, if beneficial. And for the large part, it's because of individual differences in microbiota. So one person to the next, it's really different. The composition of microbiota is very different, and this is kind of part of why different individuals have different health outcomes, but it's it's really difficult to make sure that people have the same flora composition, and it may not even be necessary for everybody to have the same composition. But in general, we know that the more diverse the flora, the healthier the person would be. We looked at the results of a specimen stain to list the most likely organisms present in the specimen. We described the basic process of microbiology testing for identification and susceptibility, and we identified the normal human flora and the role in infectious diseases. Now, let's explain the antimicrobial stewardship process for initiating, monitoring, and streamlining effective and safe antimicrobial therapy. The first thing to do in a systematic approach to infectious diseases is to start with the question, what are we treating? And there are 4 questions to ask. First, did you determine the infection site or type using subjective and objective patient information? That includes signs and symptoms that are specific to an infection type, or it could be general manifestations of infections. Second, did you assess severity using SIRS or an infection-specific tool to determine where the patient should receive care? As we learn about different infections throughout this course, we will talk about infection-specific tools that are relevant. Determining the severity of disease can help triage the patient to the appropriate setting in order to receive appropriate care. And in general, we can think of the settings as outpatient or inpatient. So if someone who is less severely ill and is ambulatory, meaning that the patient is able to walk, so the infection is not severe enough to prevent them from walking, this patient can make appointment at the clinic, make appointment with the primary care physicians, or in an ambulatory care clinic with a pharmacist in order to receive care. Now, slightly more urgent would be if the patient is still ambulatory but needs urgent treatment. So that for that reason, the patient can go to an urgent care. And then we have emergency departments. And this is kind of an overlap between outpatient and inpatient, because when people go to the emergency department, they do not necessarily get admitted to the hospital. So a lot of patients who go to the emergency department may actually get treatment and get discharged home. So they may go home with a prescription. So they will go to the pharmacy, either the outpatient pharmacy at the hospital or perhaps to the community pharmacy to receive medication, but may not necessarily require admission to the hospital. And on the other hand, some patients may be sick enough at the emergency department that would require admission to the hospital. So once that's determined that a patient needs to be admitted to the hospital, they will be sent to the general ward on the floors of the hospital. And that is where patients will receive care inside the hospital. And in severe cases, in critically ill patients, they may receive more specialized care inside the intensive care unit. So patients may actually need to go to the ICU. So that would be the most severe disease. Now, the third thing to consider is, did we obtain cultures from the infection site before starting antimicrobial agents and remove the infection source? So source control is important, because if the source of an infection is still there, doesn't matter how much antimicrobial agents you give to the patient, the infection continues to come back. So source control is important. And the reason you want to get cultures before giving antibiotics, because even a single dose of antimicrobial can pretty much eliminate organisms from the specimen. So it would be difficult for us to grow it in the culture. Even though a single dose may not necessarily treat the infection, but it would reduce the inoculum in the specimen enough that it will be difficult to have a good yield out of the specimen. And fourth, did you list the causative pathogens from most to least likely? And this is, initially, before we actually identify what's growing in the specimen, it's a guessing game. So we have to, based on the signs and symptoms and the infection, we have to guess what's the most likely pathogen so that we can target those pathogens empirically before we actually find out what's causing the infection. Now, the second thing to consider is, how can we ensure that the patient receives the most effective therapy? So let's focus on efficacy before we move on to safety. Of course, the first thing we want to do when possible is to make sure that we use reliable resources to select non-pharmacologic therapy. Then we consider reliable resources to select the antimicrobial regimen of choice. So in general, for most infections, we have first-line therapy and we have alternative therapy. So when the patient can get first therapy, that would be the best approach. And occasionally, the patient may not be able to get first-line therapy for various reasons that we will discuss, and that's when we can go to alternative therapy. And the next thing to consider, if more than one option is listed, did you use an antibiogram or another reliable source to select a regimen with activity against a high percent of likely causative pathogens listed in checklist item number 4? And here's what an antibiogram looks like. So here's an example of the antibiogram from Loma Linda University Medical Center. So here you can see that each row is a different organism. So for example, the first line of organisms is Acinetobacter baumannii. And then this, in the second column, they're showing you the number of isolates. So isolate means, pretty much how many times this organism was growing inside the microbiology laboratory. So for example, if we had, and you know, these are unique to a patient. So, you know, in other words, if we sent 2 blood samples from the same patient and, you know, E. coli grew in both, that counts as one isolate. Okay, so if you had multiple patients, so it essentially tells you over over the time period that this antibiogram is reporting. During that time period, you know, this organism was isolated in the lab 53 times, and then they're reporting you their susceptibility, the percentage susceptible to each antibiotic. So if there is a dash, that means they just did not test against that antibiotic because they're not routinely tested. So it's reported for things that are routinely tested. So for example, ceftriaxone. So when they checked susceptibility of Acinetobacter to ceftriaxone, it was susceptible 17% of the time. So that's pretty bad. Only 17% of the isolates were susceptible to ceftriaxone. Whereas when you compare that to meropenem, that's slightly better. So that's 30% of the time. And if you look across, these numbers look pretty bad because this organism is highly resistant to most antimicrobial agents. So that's why these numbers look pretty bad. Now, if you look at another organism such as Citrobacter frundii, there were 31 isolates during this time period at the micro lab. 35% of these were susceptible to ampicillin, and let's say 100% of them were susceptible to imipenem and meropenem, and 81% were susceptible to trimsulfa. Okay, so you can do the math. So this is reporting susceptibility. So let's say if 97% of Citrobacter frundii was susceptible to gentamicin, for example, that means the remaining 3% was not susceptible. So if you want to identify resistance, you basically subtract this from 100. So for amikacin, for example, the resistance rate was 0% because 100% were susceptible to amikacin. So that's how it's reported. And for some organisms, there also like report a subclass of those. So for example, E. coli, we'll talk about what ESBL means on another lecture, but some E. coli produce something called ESBL. So they're showing you that for those isolates specifically, what the susceptibility look like. And another good example is Staphylococcus aureus. In general, we like to classify Staph aureus either as MSSA or MRSA. And essentially, in the line with Staph aureus is showing you all of the isolates of Staphylococcus aureus, regardless of whether it's MSSA or MRSA. And then the next line includes MSSA only, and the next one is MRSA only, and then they show you susceptibility. So that's how an antibiogram looks like. And in general, for most times, you want numbers that are at least 80% susceptible and higher. So that would be pretty good. So these numbers under 80% are pretty bad. So if you have options, you want 80% or higher. In the ICU, when patients are critically ill, you really want 90% or higher for most infections. So that's something to keep in mind. And now you have to take that with a grain of salt. So it doesn't mean that you should only use the highest numbers. We'll talk about how to choose, because this is only one factor when we select antimicrobial agents. So once you have 80% or higher, that's good enough for most infections. You don't necessarily want to start with 100%, and we'll talk about that more on a future lecture. Now, the next thing to consider is, did you consider the active antimicrobial agents with the narrowest spectrum the drug of choice? So in general, we want to avoid a broad spectrum and focus on narrowest spectrum possible, and we'll talk about that on a later lecture. And penetration is also important. So depending on the type of infection you're treating, you want to make sure that the antimicrobial agent actually penetrates to the site. And here's what it means. So essentially, in general, we either take antimicrobials orally, so it goes in through the mouth and then it's absorbed and goes into circulation, or it's directly injected into the blood.
And there are other routes, but in general, we'll focus on intravenous and oral routes in this course. And if your infection is, for example, meningitis, you really want to make sure that antimicrobial agents you use actually penetrates into the site of infection. So it's important to know, what percentage of the drug from the blood actually goes through the cerebrospinal fluid or CSF. So that would be significant for meningitis. If you're treating a urinary tract infection like cystitis, you want to make sure that the drug actually goes to the urine. So for example, if a drug is not renally cleared, it's not going to go to the urine. So even though you get a large concentration of the drug in the blood, it's not going to go to the urine. So you want to make sure you choose something that goes into the urine. So on and so forth. So for skin and soft tissue infection, you want to make sure it penetrates the skin. For bone infections, which we call osteomyelitis, you want to make sure you get bone penetration. For peritonitis, which is in the peritoneum or peritoneal space, you want to make sure that the drug penetrates that area. Occasionally, if the infection is inside the intestines, for example, Clostridioides difficile colitis, you actually want to make sure that the drug stays in the stomach and is not absorbed into the blood. Or if you're injecting into the blood, you want to make sure that it can actually go into the intestine. Otherwise, it's not going to get to the site of infection. And for pneumonia, which is infection of the lungs, you want to make sure there is penetration into the lungs and epithelial lining fluids. So penetration is important. And for severely ill patients, you want to make sure that you consider using IV antimicrobials empirically, when appropriate. So in general, people who are severely ill, they may not have, for example, if you give them orally, they may not absorb it quickly. Or patients are sick, so they may be vomiting, so you don't want them to vomit the antibiotics out. So IV in general is initially appropriate, and of course within reason. So again, if you're treating C. diff colitis, you want to make sure that an oral route might be a better choice because the infection is inside the intestine. So you have to use these things cautiously. But in general, for most infections, IV is initially appropriate for severely ill patients. And the third thing we want to consider is we want to make sure that it's what we're giving the patient is safe. So for patients with drug allergies, you want to avoid selecting antimicrobial agents that may cause allergic reactions. If a patient is pregnant or of childbearing age, you want to avoid teratogenic agents. You want to evaluate if a patient is at high risk for adverse effects, or if there is contraindications, you want to avoid those in the patient. You always want to check for drug-drug interactions if the patient is taking other medications or over-the-counter agents, including herbals, supplements, etc. For immunocompromised and those with meningitis or endocarditis infections, we want to consider in general bactericidal antimicrobials. Now this is a bit controversial because the evidence for this is not clear, so it's not studied well. But in theory, we think that bactericidal may benefit patients who are immunocompromised or have meningitis or endocarditis. And here's what that means. So essentially, the definition of bacteriostatic means that the agent prevents the growth of bacteria, whereas bactericidal means that it actually kills bacteria. So let's look at this graph, the graph in the middle on bacteriostatic. So here on the x-axis, we're looking at time, so 2 hours, 4 hours, 6 hours, and 8 hours. And on the y-axis, we are looking at cell density measured in colony-forming units or CFU. So the first line you see is essentially the control, meaning that we have a certain bacteria, and assuming that the conditions are correct for growth, meaning there is food for the bacteria to consume and the temperature and all the conditions of the environment are correct for growth, if you leave it alone, it will grow over time, right? So 2 hours later, 4 hours later, the number of colonies will increase. And if you give antibiotics, it will essentially prevent the growth. So what bacteriostatic means, and some examples include tetracycline, erythromycin, and linezolid. If you give any of these bacteriostatic agents, it will essentially prevent the growth. Okay, so it's essentially preventing the growth. Now on the right hand side, the same thing is done with bactericidal agents. So you can see again the control line. So if you leave it alone, the colony forming units will increase over time. Now these are bactericidal, then they actually mean that they will drop the number of colonies. So it's not just a flat line. They actually decrease the number of colony forming units over time, and some of them more rapidly than others. So for example, aminoglycosides such as gentamicin are famous for rapidly dropping the number of colony forming units. So it actually kills pretty fast, and some of them kill slower. And these are all experimental in vitro. So this is not necessarily inside the patient's body, but inside a test tube where these experiments have been conducted. So the theory is that if somebody is immunocompromised and they don't have the benefit of immune system, this might be dangerous to just prevent the growth because there is no immune system to actually kill it. So you want to rely on bactericidal to kill as many of these before the bacteria kill the patient. Now, this has come under scrutiny because a systematic review of many studies actually found that for the large portion, there is no clinical difference between bacteriostatic and bactericidal agents. So in general, it doesn't matter. However, the studies, there are not good studies looking at this specifically in immunocompromised patients like transplant patients or those with endocarditis and meningitis. So some of these are based on case reports that have shown there may be benefits to bactericidal. So it's a bit controversial. In general, doesn't matter if it's bactericidal or bacteriostatic unless you're dealing with immunocompromised meningitis or endocarditis. And the next thing to consider is, if you have multiple options, you want to choose the most affordable option for the patient. Now, the fourth thing to consider is how can we ensure the patient receives the right dose? So you want to optimize the dose of antimicrobial, and this is important for us as pharmacists. So in general, you may consider a loading dose, if it applies to the antimicrobial agent, and you want to make sure that you use appropriate body weight. So sometimes some agents are dosed based on ideal body weight, sometimes adjusted body weight, and sometimes total body weight. So you want to make sure you are using the correct weight when calculating the dose. And sometimes you need dose adjustments. So depending on renal function and liver function, you may need to adjust the dose based on that. And in severely ill patients, or any time that you have MIC data available, you want to optimize PK/PD. And I'll talk about what that means in a minute. For outpatients, you want to make sure you increase adherence. So in general, it's hard to improve adherence if you give the drug more than twice a day. Ideally, once a day is best for adherence, but twice a day sometimes is difficult to avoid. But anytime you have the option between twice a day and 3 times a day, or worse, 4 times a day, you really want to do twice daily to improve adherence. And of course, always think about duration of therapy. And in general, shorter the duration of therapy, the better, because of adverse effects and unintended consequences of antimicrobial agents. So you want to choose the shortest duration of therapy that's proven to be effective. Now here's PK/PD. So pharmacokinetics in general means what the body does to the drug. So in general, you can think of, once you start giving the drug, the concentration keeps going up. And then once you stop giving the drug, and whether it was oral, so this was oral absorption, or if you were giving it intravenously, this is when you stop infusion. Then you have a drop in the concentration, so the body starts eliminating from the body. So that's pharmacokinetics. And pharmacodynamics is about what the drug does to the body. So essentially you measure the effect. So initially there is no effect. As the drug enters the body, you start to see the effect. And at some point there is like a ceiling effect. So there's only so much effect you can give, right? That's in general. When it comes to antimicrobial agents for infectious diseases, we like to combine pharmacokinetics and pharmacodynamics because they can only make sense together. So the way we think of pharmacodynamics is that we need to consider MIC, and that's MIC is the minimum inhibitory concentration of the drug. And there are some PK/PD parameters such as the ratio of maximum concentration to MIC, or the ratio of area under the concentration curve to MIC, or the time that the concentration is above the MIC. And we'll talk about those on a future lecture of when to use each of those and what they mean. But these are essentially the PK/PD parameters we use. And the reason I am emphasizing this is that there is a lot of variation or variability between patients. So there is inter-individual variability. So you can think of age, there is a difference in pharmacokinetics between a young adult and an older adult. Sometimes race and ethnicity can make a difference. Obesity can affect pharmacokinetics, comorbidities, medications, and there are also intra-individual variability. So depending on what's going on, sometimes there is altered fluid balance. If you're giving a lot of fluids to the patient, that can change pharmacokinetics. If there is renal or hepatic dysfunction, or if there is organ support. I'll talk about some of these in a minute. And then pharmacodynamic factors are things such as host immune response. So sometimes patients might be immunocompromised. Inoculum size. So sometimes, an infection can have a lot of bacteria at the site of infection. That's referred to as the inoculum size. Or sometimes there may be just a few. So the higher the inoculum size, the more difficult it is to eradicate the bacteria from the site of infection. We talked about tissue penetration at the site of infection. There are things like biofilm that we'll talk about on another lecture. And there are other things that affect pharmacodynamics. Now, when it comes to critically ill patients specifically, there are important things to consider. So, for example, there are 3 things I have listed on the left-hand side that can lead to reduced plasma concentrations. So, for example, hyperdynamic environment in the patient. So, whether naturally if the body is responding to, let's say, a drop in blood pressure due to the infection, or whether we are actually causing this by giving pharmacologic agents such as vasopressors. If the cardiac output is increased, because we want to bring the blood pressure back to normal, so the patient doesn't go into shock, or the patient is already in shock and we're trying to bring them out of shock, that can increase perfusion to other organs such as the kidneys, and can lead to increased elimination of the drug, so your plasma concentrations may be lower. So this is something to consider, which means that in the ICU you may need higher doses of the drugs. Another thing is altered fluid balance. So again, if you're giving a lot of fluids to the patients, it keeps diluting the concentration of our antimicrobial agents. So we may need to increase the dose. Sometimes there is third spacing. So if the fluids keep going into, let's say, peritoneal space or in, other third spaces, the drugs can get diluted, and so plasma concentration will be lower. The same with protein binding. So if you have drugs, for example, ceftriaxone is highly protein bound. So if there is altered protein binding, so for example, if the patient's albumin is less than 2.5, then there isn't enough albumin around to hold on to ceftriaxone, which means that unbound ceftriaxone will be eliminated from the body, so you'll have reduced plasma concentration, which means that you may need to increase the dose. And there's also the phenomenon of augmented renal clearance. This is specifically defined as creatinine clearance greater than 130. So in other words, normally pharmacies dose adjust based on renal dysfunction, right? So if the creatinine class, let's say, is less than 60 or less than 30, depending on the agent, we like to reduce the dose. This is the opposite. So augmented renal clearance essentially means the kidneys are working too well, so it's clearing the drug too much. So you actually need to dose adjust in the other direction. So you need to increase the dose because of reduced plasma concentration. And things that can lead to augmented renal clearance include things like trauma, sepsis, burns, hematologic malignancy, or pancreatitis. These are some of the risk factors, and this is more common inside the ICU. And of course, it's possible that the patient may have no organ dysfunction and be critically ill, which means that your plasma concentration are actually normal. And then on the other end of the spectrum, we have if you have increased plasma concentration. So either because of renal or hepatic dysfunction, so the drug is not getting cleared. So your plasma concentration go higher, so you need to dose adjust and reduce the dose. And sometimes there is organ support. So if there is renal replacement therapy or ECMO, you may have increased or decreased plasma concentration. For example, if someone is on renal replacement therapy, essentially the kidneys are not working, so you may have increased plasma concentration. And then also depends on the settings of the dialysis and whether it's how well it's clearing the drug. So you may have increased or decreased plasma concentration. And then things like ECMO can increase volume of distribution, and sometimes the drugs can get attached to the equipment, so it can lead to reduced plasma concentration. And here's what ECMO looks like. So ECMO is essentially kind of what dialysis is to the kidneys, ECMO is to the lungs. So dialysis, essentially the kidneys cannot work, so dialysis takes over the kidneys and it adjusts toxin, it removes toxins from the blood, and it balances fluids and electrolytes, among other things. What ECMO does is if the lungs don't work and cannot oxygenate the blood, so essentially the blood will go into this machine. The machine will exchange gas. It will exchange oxygen for CO2. So it puts oxygen into the blood and removes CO2 from the blood. And then this goes back through the pump into the body. So this is a big machine. And, as you can imagine, the blood is going through this big machine. So volume of distribution changes. But ultimately, drugs can also get stuck in this machine, so that will affect your drug levels or concentrations. And the best thing to do is to do therapeutic drug monitoring to actually, when available, because we cannot do therapeutic drug monitoring for every drug. But when available, the best thing to do is to actually measure plasma concentration and see if it's decreased or increased, and make adjustments based on that. For example, a good example is vancomycin, where we can get plasma concentrations, and based on the level, we decide whether we should increase the dose or decrease the dose or leave it untouched. The last thing that we should consider is how can we ensure this works and does not happen again. So we do want to specify how and how often we'll monitor for efficacy and safety. If cultures and susceptibility results have returned, you do want to go back to number 8 and start from number 8 in order to optimize the dose and regimen. And did you recommend immunizations and other preventive measures? So that's important. And lastly, as pharmacists, you do want to provide counseling, not just about the antimicrobial agents, but also about the infection. So what to expect from the infection, how often, what to look for, and what to expect as far as resolution of signs and symptoms, and of course what to expect of antimicrobial agents adverse effect profile, how to take it with food, with water, without food, or and how long to take this. This concludes this presentation.